So we have done production of haploids by anther, and then we also did by pollen culture. Now we'll do by ovule culture. Okay, this is known as gynogenesis. All right, when you synthesize a plant via your ovule, also is known as gynogenesis. So we can develop haploid plants from the ovary or the ovule cultures, because the ovule is having only one set of chromosomes. It is only N. Okay. We can trigger female gametophytes or the megaspores to develop into the sporophyte. Okay, gametophyte is the plant body which is only N, and sporophyte which is 2N. Okay, so we can develop we can trigger the female gametophyte, that is the megaspores. You remember, we will get 4 megaspores during after meiosis 2, and then 3 they will degenerate. Okay, so any of these they can use to develop the sporophyte or the plant body. So gynogenic haploids first developed by Sam Noem from the ovary of Hordeum vulgare. What is this Hordeum vulgare? I've forgotten. I forgot. So it was later applied for rice, wheat, maize, sunflower, sugar beet, and tobacco. I remember now, Hordeum vulgare is barley. Okay, barley. This one. Okay. So they applied it to other food crops like rice, like wheat, maize, sunflower, sugar beet, and tobacco. Okay, because barley is your just like your rice and wheat. So we can culture them in the lab, so in vitro culture of the ovaries which were not pollinated. Okay, still in the N. Okay, still in the N number of chromosomes, still having only half the genotype. So we use these when we cannot achieve the haploid plants via the anther culture. So we will go for the ovary culture. So let's look at the process. So you take again the young flower buds, okay, still a closed one. No, this is the calyx, this is the corolla. No, still closed. Don't go for a open flower, because open flower means the style has received so many pollens. No, so again go for the flower buds. At least 24 to 48 hours before 1 minute, let me remove that, before the anthesis from unpollinated ovaries. Okay, prior to anthesis, what does that mean? Okay, after removing the calyx, corolla, and the stamens, the ovaries are subject to surface sterilization. So again we'll use sodium hypochlorite. Remember that it's like a salt, hypochlorite. So you surface sterilize it, then you cut the ovary at the distal part of the pedicel, is inserted in the solid medium. So if this is the pedicel, okay, pedicel is the stalk, remember? Like if this is the leaf, this one is the pedicel, okay? So you cut it from here where the stalk attaches, and then you insert this attached cut part into the culture medium. So this is just the parts of the flower, the diagram of parts of the flower. So whenever liquid medium is used, the ovaries are placed on a filter paper or allowed to float over the medium with the pedicel inserted through the filter paper. So here, if you decide you don't want a semi-solid media, you like it to just be free floating, then you just insert the pedicel part into a filter paper. Okay, because you have solid media also, you have semi-solid media also, you have liquid media also. So for these very delicate parts of a flower, no, which are so small, like a leaf, yes, fine, you insert it in a semi-solid. But something as small as an ovule, I'm talking just about that the egg only, something so delicate. So you might have to use liquid medium. Okay. Okay, let's go on. The commonly used media are, the most common is the MS media, White's media, niche media, N6 media. Then production of the gynogenic haploids is useful in plants with the male sterile genotype. Uh, uh, um, there are some flowers, where the males are already, remember girls, the self sterility chapter. If this one is S1 and this is S2, okay, the ovule is of S1 S2 genotype, and this obviously the anthers will be of S1 S2 genotype. This is also S1 S2 genotype. Even if by chance this one lands here, this pollen land here, it cannot go in. Okay, it's already blocked. But let's see another free floating one which came via the air or the bees, which is a different genotype. Anything other than S1 S2, take a wild guess. Let's say S7 S4, whatever. It can germinate. Okay. So the self-sterile ones are easy. I mean, the male sterile ones are easier. So let's look at a few of the limitations of ovary or ovule culture. The dissection of unfertilized ovary and ovules is rather difficult. Well, you girls, you know the most. You know better than Miss also. You know better than Miss Amy also, because most of the dissection of the ovary and ovules you have been doing, all these years. So you know how difficult it is to, dissect the ovaries, and to take out just the ovules. Just imagine even how more difficult. And then how many ovaries are there in one flower? There's just one. You need only one, because the three, they degenerate, so you have only one chance. So that is also another disadvantage. So how to identify the haploids? You can go through the morphological approach, the genetic approach. Okay, let's look at the morphological approach. The floral parts and the cell size of haploid plants are smaller and reduced when we compare to their diploid counterparts. But by looking only, it's still very difficult to say. So the genetic approach will be, we can have some genetic markers. A1 marker for brown colored allurone, A marker for purple color, LG marker for ligule-less character. The above markers are used for the development of haploids of maize. Okay. So when you are doing molecular work, so you have conducted the experiment. Okay, you have loaded here in the wells, you have loaded the DNA, and then you ran the electricity, here anode and cathode. So since DNA there's a charge character, it has a negative charge, so it will tend to travel from positive to negative, from negative to positive. So as it goes, it will form bands, as it travels, because as you go higher, this thickness increases of the gel. Okay, so there are some markers, okay, which will always show. So if the allurone is purple, means we got a haploid plant. Okay, so then let's say here the DNA from this plant number 3, it didn't show any marker, but DNA from plant 1, it had a marker for ligulous character LG, and the DNA from the plant that we extracted of the plant 2, okay, the plant 2, this is plant 1, this is plant 3, DNA from plant 2, it showed for allurone, what was that, A or something, the marker for allurone, it showed clearly, it was a dark band. So you know that this plant of P2 and P1, they are haploids. Okay, so I just gave you a very rough idea of molecular, so that you'll understand. So after creating haploid plan, no, to make it strong, you have to diploidize it. Okay, make those n into twice n. Okay, so you got your haploid plants by androgenesis or gynogenesis. These plants, they may grow up to flowering stage, but the viable gametes cannot be formed due to lack of one set of homologous chromosomes. Okay, so there will not be any seed formation. So fine, you have created the haploid plant. Fine, you have grown. Fine, everything was fine. But it can flower, yes, but how can the gametes be viable? Because it has only one set of chromosomes. So you have to diploidize. You have to double the amount of chromosomes of the haploid plants. So you can diploidize them by uh, some chemicals. Okay, so there are two main approaches. First, you can do it by colchicine treatment. We had seen that in the androgenesis, remember? And you can do it by endomitosis. Okay, so you have to diploidize it, or you have to duplicate the chromosome. So colchicine is a chemical which is the most commonly used for diploidization of homologous chromosome. Okay, it inhibits the spindle formation during chromosome duplication. Okay, do you remember in mitosis? Only you and Miss RL will know this. Okay, which stage duplication of mitosis occurs? And I know Miss has been teaching you again and again, so you revise her topic also. So during the spindle formation, no, this colchicine will bind with those spindle fibers and it will block. Okay, so that those spindle cannot pull apart. Okay, to the different poles. So let's look at how they do it. So when the plants mature, colchicine in the form of a paste is applied to the axil of the leaves. Now the main axis is decapitated. This stimulates the axillary bud to grow into diploid and fertile branches. So you just take a leaf, this one, no, this is a leaf, okay. You just cut this part. So you have cut, okay, you have cut this one, you have cut it. So this one now, since it is a budding, it's a bud, no, it's budding, it gets confused. So you have applied that chemical, no, this one which will you apply the colchicine. Then this part, whether it becomes branch or becomes leaf, it will become 2n. Okay, the rest is n, but here the rest of the bud is twice n because of the colchicine. Then you can treat directly the young plantlets with colchicine solution, or you can just treat the axillary buds with colchicine. Okay, means the young buds, okay, just before they start to grow, you keep adding here colchicine, then they will become diploid. What happened? Okay, is okay. Then you can go with another method, which is endomitosis. It is a doubling of a number of chromosomes without division of the nucleus. So in general, the haploid cells are unstable in culture and then with a tendency to undergo endomitosis. So it is in its own nature there. How do I say? It's like a habit of chromosomes. It's the habit of a chromosome to always be paired. Okay, it is a habit. It's very unstable. Just one of this and one of that is very unstable. So it is habituated to double itself naturally. Okay, simply it will just duplicate itself to always be in a paired stage. Okay, so this endomitosis is you can exploit it, you know, this habituated nature of this chromosomes. Like let's say some of you keep walking home, right? So one day you met a friend and then your friend said, let's go to your house, and then you chat the whole way. You don't realize, but you already reach your house without realizing whether you took left or you took right, but your feet already knows. It's habituated to walk this particular direction to reach your house. So that also this nature of your chromosomes. They like to be in pairs. So let's look how to do it. So you grow a small segment of a haploid plant stem in a suitable medium and you add the growth regulators. Then this induces the callus formation followed by differentiation. Then during the growth of callus, chromosome doubling occurs by endomitosis. Okay, because callus is nothing but cell. This cell mass, it has a habit of being stabilizing itself. Okay, so it will always double. So with this chapter is complete. Thank you.